Okay, thank you and welcome to our second review of Bioscience. This review is for the final exam. And thank you and welcome everybody. Today is Saturday 10 December 2016. So let's start our first for our review. All right, so the first lecture, the lecture 11 is going to be mostly talking about DNA, cell division, and aging. So what I want you to remember from all this material is what I'm going to remark at this moment. So number one, and please take note about this, we have the DNA. The DNA properties, that is the first thing we are going to check. Is replicating itself, is going to direct synthesis of proteins, so you know that transcription, right? And they can mutate spontaneously and going to store information. So that is the properties of DNA that we must remember. So about histones, the histones are just proteins, as you see here, where the DNA is going to coil around, This means that the purpose of this is to organize systematically the DNA and not to get tangled. So that is about what is doing the histones. Okay. So here we have the histone. Can you see here the histone? This green stuff is a protein, and these are actually the chain of DNA. So in that way, the DNA is not going to get tangled. So that is the only reason, well, that is important reason for about the histones. All right, so talking about cell division. In the cell division, what I want you to remember always about cell division is that there is all cells in the body are going to have cell division. Some cells are going to divide faster than others, especially the ones who are going to be faster are the ones who can be affected for some chemistries, like for example chemotherapy. Fast cell division is going to be, for example, the hair that is going to be the intestines, the lining of the intestines, the bronchial tree, the lining of the bronchi. So those are the one who has fast division. All right, so let's keep going. You have your practice questions. We have the DNA replication. You already know what is the chromosome and the centromere. The centromere is this portion of the chromosome that are going to have some activity. Look at this. This is the centromere. This centromere is going to be divided and multiplied during the cell division. Centromere is like the holding all the branches of the chromosome. And what we have here in the centromere are DNA. Those are we call junk information, but it's not that really junk information because this DNA that we have in the centromere having some activity to produce some protection to the other part of the DNA. So here we have a centromere. So we clear here, centromere is divided during the cell division. All right, so another thing that we need to remember about this is the centromere, the telomeres, and the telomeres are the tips of the chromosome, chromosomes. This, in addition to that, they are going to have information that are not important. They are not going to have important proteins to produce, but it's known that the telomeres with the time, when they replicate the cells, so your cells are not the same all the time. They are going to change during the years. So your cells now, the cells like 3 years ago, 4 years ago are not the same. All are being changed. And when they divide, the telomeres are getting smaller and smaller. What are the telomeres? The telomeres are the distal portions of the chromosome, this and this. So these are distal portions. And with the time, when they divide for a new cell, new chromosome, they are going to be shorter and shorter and shorter and shorter. So that is related to the aging process of the individual. Okay, so one of the things that are going to make the telomeres to get shorter faster is the stress. One thing I want you to remember about this part is that cortisol is the stress hormone. Cortisol is the stress hormone. Just give me a second. Okay, so stress hormone is going to be the cortisol. Remember always this. We are going to see that a lot. So cortisol is going to be the stress hormone, and when you have too much stress, you get older faster, right? So that's why it's going to be a more, it's going to get shorter in relation with the telomeres. Okay, let me see here. Okay. Sorry, I need to just make the ink here as kind of. Okay, all right, so let's keep going. All right, so that is about that. Metastasis is, we can see that is the invasion of cancer cells, the invasion. So you remember, and that is something I've been asking before, and actually I want you to remember, the cancer cells are having very fast division, and don't forget that, it's important. So very fast division. And there is, of course, more aggressive cancers and less aggressive cancers. But what the concept I want you to remember about metastasis is the cell invasion of the cancer cells, and that they are very fast division. All right, so talking about the cell division, you just remember the steps. We have the prophase, we have the metaphase, we have the anaphase, and we have the telophase. So ana, meta, prometa, ana, telophase. Okay. All right, so let's finish this part of the cellular division and all that. So, and let's go to our topic, skin and skin disorders. Very important for the final exam. So, do you remember, and you, in the previous lecture, we was talking about the first line of defense, remember? So, this is way back from lecture 11. So, the first line of defense is going to be the skin. And here we have in the skin, we are going to have the normal flora. Don't forget about the normal flora. The normal flora in the skin is going to protect against infections. All right, so we have the first layer, that is we have the epidermis and the dermis. Those are the layers of the skin. Hypodermis is under the dermis, and you will see that is actually formed by loose fat. So this is, if you see here, the epidermis, the upper layer. Here we have the dermis, and below we have the hypodermis, below the dermis. The skin is going to be conformed by epidermis and dermis. All right, so we have here, if you remember here in the epidermis, we have the keratin cells. Keratin is a protein, by the way, keratin is a protein that is going to be conforming the epithelial cells. Okay, the keratin. And the keratin, what we have in the keratin is the melanocytes, right? Melanocytes. The melanocytes have the melanin. Melanin is what is going to give you the color of the skin, right? So that's it, what we have in the epidermis. So epidermis, we have we have about 5 layers of epidermis. So 1, 2, 3, 4, 5, 6, 7. So these are the 7 layers that are going to change according to the age, but that is another topic. But here, what I want you to remember is that we have the stratum corneus, stratum cornea, stratum corneus or cornea, corneus, that is the outer layer of the skin. That is what is going to slough off and replaced by the layer that is coming below. All right, so melanin, melanocytes, and the stratocorneum. These stratocorneum are dead cells. They are not alive cells. And the function of the skin is to be the first line of defense. Below this, you have all the, sorry, 1, 2, 3, 4, 5, 6, 7 layers here. So all from here to here are going to be the epidermis. So they have up and downs. Can you see here? Up and downs, up and downs, up and downs. And that is all, this is the epidermis. Below that, we have many structures, right? The dermis. In the dermis, we have B, vein, we have A, we have arteries, and we have N, we have nerves, right? Besides that, we have many other structures. For example, the dermis is coming here from all this structure. We have nerves, this is the receptors for pain, for cold. Then we have arteries, we have veins. We have here in this part, we have the hair, the hair follicle that is in the dermis. Here we have the sebaceous gland that is in relation with the follicle to produce this oily appearance of the hair when you don't have shower for a few days. They are going to have hair, some oily appearance. Okay. Below that, we have, in addition to that, we have the sweat glands. We have the sweat glands that are going to just release the fluid with electrolytes. All right, so we have hypodermis. The hypodermis is this portion that is, we are going to have some loose fat tissue, and that is going to be the transition between the skin and the muscle. Here we have the muscle. This is the muscle. So hypodermis is just loose fat that are going to be the transition between the skin and the muscles. Okay. So that is about the functions of the skin. Another important topic for final is, first of all, the skin protection of organisms from microbes and dehydration, right? We was talking about that. So the skin is not going to allow to lose water from the body, and it's not going to make water passing through the skin. So it's actually impermeable, and that is going to prevent the dehydration. We have lower regulation of temperature, high temperatures. What happened with your vessels? What happened with your vessels when you have high temperature? Your vessels normally are this size, but when you have heat, that is going to turn into big vessels, vasodilation. This vasodilation is going to make closer to the skin, and that is going to make you lose water, heat, because at the same time you are sweating, and the water, when evaporate, that are going to take out the heat from the body. So that is one thing, the hydration. So protect the organism from microbes and dehydration. Okay, it's like a capsule for us, are going to regulate the temperature. Another thing is going to collect the sensory information. So we have pain receptors. Remember the efferent and the efferent. Okay, so the efferent is going to be in contact with the pain receptors. Thermoreceptors, the same thing. They are going to mostly, when you have heat, you can actually have a pain too. So their association of the thermoreceptor with the pain receptors. Pressure receptors, sensory receptors that are going to make you feel what you are touching. If you're touching, for example, can you imagine try to eat with a spoon and you cannot feel the spoon, right? So that is the sensory receptor, and that's very important. So we have collection of sensory information. So that is what I want you to keep in mind, the sensory information. Then production of vitamin D. Vitamin D, don't forget the vitamin D. What do we need in order to produce vitamin D? We need cholesterol. Cholesterol is needed in order to produce vitamin D. This vitamin D is not active, so it's going to be produced by the skin, but in the skin is not active yet, not active yet. So where they're going to be activated are going to be activated in the kidney, okay, in the kidney. So that is what I want you to remember about the vitamin D. We have excretion of electrolytes, when you are sweating, okay, you can have some electrolyte imbalance because of that, electrolyte imbalance. Why? Because when you are sweating, you are losing water and you are losing sodium. But what happens with the sweating? When you sweat excessively, you are going to lose more water than sodium. And what happened? You're going to have some high concentration of sodium in the blood that is going to be dangerous for your functions. All right, so excretion of electrolytes, through the skin, through the sweating. Absorption of some fat-soluble substance. For example, they are going to be, what are fat-soluble substance? Fat-soluble are going to be the vitamin A, vitamin D, vitamin E, and vitamin K. So these vitamins are fat-soluble, so they can actually pass through the skin easily than for example, vitamin B and vitamin C. They are water soluble. B and C are water soluble, so they are not really easy to pass through the skin. The ones who pass through the skin easily are going to be the FADEC, the A, D, E, and K vitamin. Okay, so let's keep going to the skin, the cubitus ulcers. They are going to have 4 stages: 1, 2, 3, and 4. The cubitus. The cubitus, what is the cubitus? The cubitus is when the patient, this is the head, this is the nose, this is the body, and this is your feet. So that is the cubitus position. When this cubitus position, what happened is this patient, the back and the back, the back and the behind, they are going to be in contact with the bed. This is going to have pressure. They are going to have a pressure. This pressure are going to make the circulation in this area, especially on the lower back, are going to produce some local ischemia, and the tissue start to become ischemic, and they can produce ulcers. Okay, ulcers. All right, so that's what we call bed sore ulcers. The most common is the sacral in the sacrum, and we have on the feet. On the feet, we have actually the most common bed sores. All right, so we have stage 1 and stage 2. We have here that the stage 1, we have stages are going to be on how deep is going to be the ulcer. Stage 1 is we have the epidermis is going to be involved only, epidermis, so the outer layer of the skin. We have stage 2, so 1 is 1 layer, epidermis 1, stage 1. Stage 2 is 2 layers. We have epidermis and dermis. Then stage 3 is going to involve the muscle inclusive. So epidermis, we have the dermis, the hypodermis already, and all the subcutaneous tissue. Hypodermis, by the way, hypodermis is the same to say subcutaneous tissue. Subcutaneous tissue. Subcutaneous tissue, sub means below the skin. Hypo means below the skin too. Subcutaneous tissue and subcutaneous tissue are going to be the same. So stage 3 is going to involve all these 3 layers, including the part of the superficial portion of the muscle. So we have epidermis, dermis, and hypodermis, and start to be the muscle being involved. Stage 4, they are going to involve all the layers of the tissue down to the bone. So they are going to include epidermis, dermis, hypodermis. We have what else? The muscle, and we have the bone. Okay, so bone 4 is the more severe, the more deeper. Okay, talking about the burns. Burns, we have superficial first degree, we have second degree, and third degree. So three things. The first degree is going to be just redness. The second degree is going to be like vesicles. So when you have vesicles pumping with fluid inside, vesicles, what is the elevation of the skin with fluid content. So that is actually a vesicle. When you have that redness, or we're seeing redness, okay, is going to be second degree. And third degree is when you have actually the burn is going to appear these necrotic areas. Can you see these black areas? So those are necrotic. Necrosis means dead tissue. So dead tissue need to be removed. Why? Because if you leave it there, they're going to create bacteria. So it's a source of infection. All right, so let's keep going. Oh, by the way, math, there is no math in the exam. All right, so let's keep going with sexual reproduction. We already know sexual reproduction. I'm going to make a small summary here about that. So let me create a page, please. Just a moment. Uh, okay. Okay, so here we are going to see that we have sexual and oh my God. Okay. Just a moment, please. All right. So here we are going to see the sexual. We have the sexual and asexual. Okay, so let's make a very brief summary. The sexual, we have two gametes. The gametes can be the ovum, it can be the sperm, right? So are sexual cells. And asexual are the rest of the body. So the sexual, mostly what we are going to have is meiosis, and asexual is going to be the mitosis. Okay, in meiosis you have the mother cell 2N, and mitosis you have 2N. Meiosis you're going to have two mitoses, consecutive mitosis. So at the end what we are going to have is four daughter cells, and the mitosis we have two daughter cells. Here we have two N chromosomes, sorry. We have N chromosome, N, and N chromosome. These two N are going to be called diploid. And this N half is going to be haploid. And this coming from a diploid cell, right? But the resultant are going to be the haploid cells. So that is, and you remember the prophase, metaphase, anaphase, telophase of mitosis. We have double mitosis here. We have prophase, metaphase, anaphase, and again, prophase, metaphase, anaphase in meiosis. So the resultant is going to be the spermatozoid here, sperm, or the ovum. In the sperm, in male, they are going to give the tail later, right? In female, they are not going to have it. So it's just the N chromosome, the N cell. All right, so that is about the process. Sorry, let's go back here. The process to produce sperm, and the process to produce ovums. The process to form sperm is called spermatogenesis. And ovum is the like that, oogenesis. Okay? All right. So, but all together is going to be called the gameto, because Both are gametes, right? Gametogenesis. So that is what I want please to always remember, okay? So gametogenesis produces sperm ovum, the process of sperm is spermatogenesis and ovum oogenesis. All right, so let's go over the homologous chromosomes, and we already know, for example, if you have, this is the father and this is the mother, if you compare chromosome 1, if you compare with chromosome 1 of the mother, that are homologous, okay? So if you compare chromosome 7 of the father or mother with chromosome, you need to compare with the chromosome 7 of the father or mother. So same chromosome comparison that are going to be homologous. But if you compare 1 to 7, these chromosomes in comparison are going to be heterologous. Why is important about this? Because if you have, for example, your color of your hair is in chromosome 1, okay? In chromosome, the color of the hair of the mother should be in chromosome 1, 2. So when you are talking about homologous chromosomes, you are trying to say that you're comparing characteristics from father and mother. You cannot compare the color of the hair with the size of your foot, for example, right? So that is going to be just same characteristics comparison. That is when you're talking about homologous chromosomes. And this is homologous chromosome. And then we have another name that is called the homozygous. Remember, homozygous and heterozygous. Heterozygous. So these two are different from these homologous and chromosomes, okay? Homologous and heterologous. So homologous and heterologous are going to be this, what I just explained. Homozygous is when an homologous chromosome from father and mother, they have, for example, green eyes and green eyes. So these homologous chromosomes, they are homozygous, because they have the same characteristics. Zygous means egg, okay? So homozygous, and when you have heterozygous, you are comparing homologous chromosomes of color of the hair, for example, color of the hair, but the father is green eyes and the mother is brown eyes. So that means that are going to be heterozygous. All right, locus, is the location, and allele. So again, let's see here. If we have a chromosome here, we have a chromosome, and we have another chromosome here. So what happened is this is the color of the hair of the father. This is the color of the hair of the mother. So I didn't put it here. I didn't put it here. I didn't put it here. I put it here. Equally equivalent from father and mother. Those are actually, we call this alone and locus. This is the location of this chromosome. In this chromosome, locus. In here, we are going to see the location of this gene, in what we call locus. Now, locus. Here, when you compare these chromosomes that are going to be called allele. So those are allele. Okay, allele. All right, so that is about that. Genotype. Genotype is the distribution of how the gene is being made. So something that you cannot see, right? The genes with the nucleotides, with all the components of the genes are going to the recipe for green eyes, right? The recipe for brown eyes. So that is the genotype, the recipe, and the phenotype is the result of that cooking. Let's put it this way. So this is something that you can see. Okay. All right. So trait is both of them, genotype and phenotype. So trait is going to be as a consequence of the recipe and what the result of the cooking. All right. All right, so we have a very important thing here is about, sorry, I'm using mouse and something else that is making difficult to manipulate the. Okay, let me see if I have some questions. No, not so far. Okay, let me take down. Okay, so here, let me see if I, oh, this is better. Fine. Okay, so here we have the autosomal and the sexual chromosome. So autosomal and sexual. Okay, autosomal are going to be 1, 2, 3, 4, up to 22. So all these guys are going to be autosomal chromosomes, autosomal. And the sexual chromosomes are going to be these Y and X. So in this case, this karyotype is for male, right? X and Y, X and Y. All right, so that is what it is, the chromosome. So one side here is from the father, and the other side is from the mother, or vice versa. So they compare the gender, the chromosome coming from the father and the other from the mother. All right, so that is about meiosis. So you can tell here, we have the cell division, the prophase. Then are going to be the metaphase. They are going to be in the equatorial line. They are going to start dividing. This is the anaphase. Can you see the centrioles? The centrioles are going to lace the chromosomes. So this multiplied like for 23 times. Okay, so but here's just a cartoon. They are trying to just demonstrate how are the functions of this case is the centrioles. And the centrioles are different from centromeres. Okay, centromeres is different. Where is the centromere? Centromere is this. Remember, we have a chromosome. The centromere is this. So it's different from centrioles. Centromere are going to be attached to the centrioles attached to the centromeres, and they're going to pull it. That is the anaphase. Then we have the telophase, right? The telophase. And then again, because I said meiosis is going to repeat the process again twice. So what is the result? We have 4 daughter cells, right? We have 1, 2, 3, 4. And how many we have? N chromosomes each. And how we call this? Haploid cells. It can happen in female, it can happen in male. In female is going to be like this, 4 ovules, and in male is going to have the tail, well, it's bigger, and that is going to be the spermatozoids. Okay. All right, so here we have the gamete. So this is the landing, touchdown of the spermatozoid on the ovum. Okay, so this is the touchdown, and what happened is nobody else is able to get into the. When they touch down, they trigger out the reactions on the cell wall of the ovum that is going to completely close the entry to another spermatozoid. Sometimes they're going to touch down two at the same time. So that is going to create twins. So gamete, you already know what is a gamete. Gamete is a sperm, or is going to be called an ovum, right? Depends what we are talking. Gamete is the big umbrella for this. Fertilization is the fusion of two gametes. The fusion of two gametes. So we have the spermatozoid and we have the ovum. That is the fertilization. And the fertilization mostly happen in the fallopian tube. Where is it going to happen? In the fallopian tube. Okay? So the fertilization, the fusion, what is going to do, the spermatozoid plus the ovum are going to lead into what we call the zygote. Zygote or egg is the same. Egg or zygote are the same. Okay. So how many chromosomes we have in the spermatozoid? N. How many chromosomes we have in the ovum? We have N. Chromosome N. So we have N plus N is 2N. So that is going to give a zygote with 2N chromosomes, 46 chromosomes. And they are going to start dividing and produce our system. Embryogenesis, again, here we are going to see that is the for embryo, your good in medical terminology. Genesis is the origin, So it's the origin of the embryo, and the embryo is going to be up to 8 weeks of gestation. GA means gestational age. W weeks, that is the shorthand for what I said. 8 weeks of gestational age. Okay, gestational age. So that is about more than 8 weeks is going to be called fetus. More than 8 weeks, this is less than, equal or less than 8 weeks. More than 8 weeks is going to be called fetus. Less than 8 weeks is going to be called embryo. Okay. Okay, so here we have, well, he's already here. First 8 weeks, embryo. Fetus is after that. Gender is going to be determined at the time of the fertilization. At the time of the fertilization, the gender is going to be already established. Okay, at 7 weeks, they start to see from outside of the body, the genitals are start to form. So you cannot see until 12 weeks of gestation by ultrasound. That is the earliest that you can see gender. So after 12 weeks, it's going to be 3 months. So after 3 months, you can tell the gender. All right, so let's keep going. Meiotic error, so we are going to just pass this. We have some terms that we need to remember for is this. So we have our cells are diploid. So that means that is a normal cell. A normal cell is equal to say euploidy. Euploidy with Y. Euploidy. Normal cell. Diploid is a normal number of chromosomes we have in our body, right? So normal is called euploidy. Euploidy, when is abnormal, is called the aneuploidy. Aneuploidy. And under aneuploidy, we are going to have the monosomy, trisomy, because monosomy means one, right? So it's lack of one chromosome. Trisomy means an excess, it's an addition, one chromosome. Trisomy and monosomy. So those are under aneuploidy. Aneuploidy. What is aneuploidy? Abnormal number of chromosomes. For example, this monosomy, we are going to have 45X0. That is, for example, the Turner syndrome, one syndrome that Turner syndrome. So in this patient, they don't have 46, they have 45. Why? Because they lack one of the sexual chromosomes. So she or he is not X, is not Y. Okay? And trisomy is because you have an excess of chromosomes, T21. So in the chromosome 21, instead to have one chromosome 21 from the father and one chromosome from the mother, they are going to have an extra chromosome. Remember the karyotype? In chromosome 21, instead to have 2, we have 3. So that is trisomy. That is 47 chromosomes already, right? So that is in the under aneuploidy. That is an abnormal number. So, all right, so let's keep going. Okay, so we have the X-linked genes. Talking about the X-links, so you already know that the X-links. So we have the color blindness, right? Color blindness, and the color blindness is mostly related to chromosome X, right? So let's suppose that this is the abnormality, but in female, when you have another X, this portion that is abnormal is going to be compensated by the other portion of the X. We have another X in female. So they are going to compensate and make the disease do not appear. So it's a compensation. Meantime, in male, if you have the chromosome abnormal here, the gene abnormal in this portion of the X, you have Y. So you cannot compensate. And that is about, for example, color blindness. Color blindness, remember, color blindness. And we was talking about hemophilia, okay, hemophilia. So those are actually diseases related to sexual chromosomes, All right, so let's go now to the reproductive system into the male, okay. All right, so here we have high yield topics. Number one, I want you always remember gonads. Gonads are going to be the testicles. It's called testicle or testes, is the same. And the other is the ovaries, ovaries, okay? So that is the gonads. Gonads, when you say gonads, you can see either way, whatever. Then we have the gonads, that is the testicle, and the accessory reproductive organs that we are going to see. Here is the seminal vesicle. Where is the seminal vesicle? Let me see if I have a picture here. Better, yeah, okay. So here's the testicle. Let's review very quickly. Testicle. Here we have the testicle, where is going the spermatozoid. The spermatozoid is going to be formed where? In the seminiferous tubule. Remember the seminiferous tubules? Let me see. All right, there you are. Seminiferous tubules, that is inside the testicle, right? From the seminiferous tubules, they are going to run through certain structures called the striae, retetestis, whatever, but they are going to go into the epididymis, this guy. So the spermatozoid is coming this way, here, 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 and they are going to get here in the epididymis. These spermatozoids are going to go to the epididymis to get maturity. Because these sperms, even though they have the tail, but they don't know what to do. So they are totally immature. They are going to stay in the epididymis just waiting for a certain time in order to get mature. What is going to get mature is going to all the enzymes that are in the cell of the on the cell of the spermatozoid. So this is go from the seminiferous tubules, go to the epididymis. From the epididymis, they go all the way here. So this is the entry, and they are going to come all the way down, and they are going to go to the vas deferens. Remember the vas deferens? The vas deferens. The vas deferens. So that is the pathway. So it's going to go here, then down, and then go up, and that is the vas deferens. So if you see here again, we have the testicle, we have epididymis, we have the sperm is coming this way, go to the epididymis, they take a break there, they're coming back, and they are going to go all the way to the vas deferens, okay, vas deferens, see, it's getting from the scrotum. Remember the scrotum is not testicle, the scrotum is the membrane who covers the testicle, and the scrotum, the scrotum containing the testicles is from outside of the body. And this vas deferens is going to start getting from outside to inside. And if you remember the inguinal canal. And then they're going to go past all the way up, and here is where it's going to join. So even though at the level of the urinary bladder, they're coming back here, and they are going to find the seminal vesicle. This guy, seminal vesicle, is going to have, remember, is going to have fructose, right? Fructose. Okay, so I'm going to write it down here. Fructose. Fructose is a monosaccharide, right? That is going to give nutrients to the poor spermatozoids that they need to run very long way until they go into the fallopian tubes. Okay, so here we have the seminal vesicle fluid, and this seminal vesicle fluid represents about 60% of all the semen. 60% about all the semen. This seminal vesicle fluid are going to, if you see here, are coming this way, and is going to find together with the vas deferens. So the vas deferens are going to join here, is going to join here, and the other seminal vesicle join together. And this portion, can you see this portion? That is going to be the ejaculatory duct. The ejaculatory duct. So this ejaculatory duct joins the urethra. This is the urethra. This is called the prostatic urethra, and this is the penile urethra. But this, because it's passing through the prostate, that is called the prostatic urethra. Look at this, where the location of the prostate is under the urinary bladder. This prostatic fluid is going to contain about 30% of the fluid, and they are going to be alkaline, very alkaline. The reason of that, if you already know, that they're going to arrive to an environment that is very acid and it's kind of very hostile environment. So what they are doing is to neutralize the acidity of the area with the alkalinity of the prostatic gland. Okay, so here we have a sperm. This is the spermatozoa or spermatozoid. Semen is the spermatozoa or sperm plus the secretion of 3 glands. So semen is going to be the semen is going to be the sperm plus seminal vesicle fluid plus prostatic fluid plus the Cooper's gland fluid. Copper gland fluid. Cooper gland fluid, this copper is a very clear fluid that is going to help for lubrication. Okay, so those are the three things. All right, so let's keep going. Remember, the spermatozoids are produced in the what? In the seminiferous tube. So we have here, we have the prostate gland, prostatic fluid, seminal vesicle fluid, the vulvo-urethral gland that is actually what we call the litre, but we don't care about that. Just remember about this, please. This is a question, by the way. Acidity is going to be the prostate, right? So to neutralize acidity. So, but remember again, what I said is sperm plus seminal vesicle plus prostatic fluid plus the Cooper's gland fluid. Those are called the semen. Okay, sperm is not semen. Okay, sperm is just the spermatozoid. All right, so let's go to the fallopian tubes. Talking about the fallopian tubes, we have very important to know. We have that is going to be formed by the uterus plus the adnexa. Please, adnexa. Remember that adnexa. What is the adnexa? Adnexa are ovaries and the fallopian tubes. So what is the reproductive system? Uterus plus adnexa. All these two are the adnexa. So when you said adnexa, you are talking about, it's a big umbrella for ovaries and fallopian tubes. Okay? All right, so here we have the uterine division. It's going to be, this is the cervix. Can you see the cervix? The cervix is the size of a marshmallow, by the way. It's 3 by 3 by 3. And all is like a cube. It's going to be like a cube. All the sides are going to be similar, same size. So that is the cervix. Then we have here can you see an arrow? This is like a waist of the uterus. This is the end of the cervix. Then it's going to continue with the body of the uterus. That is the more prominent or more largest portion of the uterus, and we have in the fundus, right? Very suggestive name is the fundus of the uterus. It's going to be at the bottom of the uterus. So knowing these three portions of the uterus, we need to remember that this cavity is just a drawing situation because the uterus is a virtual space. It's a virtual space. So what means virtual? This is a real space. If you cut the uterus in half, you will not see that. So this is not a space. The uterus is going to be a virtual space. It's going to be like overlapping the walls of the uterus. Anyhow, so here we are going to have 3 layers, right? 3 layers. The inner layer, we have the middle layer, and the outer layer. So 3 layers. This inner layer is going to be the endometrium. You're good in medical terminology. I hope everybody finished already. Third review. And here we have the myometrium. The myometrium. Myometrium is, myo means, well, it's muscle. We know that. And we have here the perimetrium. This is the perimetrium. Perimetrium is the outer layer of the uterus. So we have already what is coming, what is slough off every 28 days or every month are going to be the endometrium. The endometrium is going to come out, is going to be bleeding about 80 cc per each cycle, and they are going to be replaced again for new endometrium. So every cycle is going to happen that for about, as we mentioned, about 525 times in your span life. Okay, so here we have the fallopian tubes. Fallopian tube, this is the fallopian tube. Fallopian tube, you already measure your fingers, right? So your fingers about 8 centimeters. Just calculate that this is about 12 centimeters. 12 centimeters. 12 centimeters. So this ovary is going to be size 3 by 2 by 2. 3 by 2 by 2 is like the size of your nail. Okay, so that is the nail of your thumb. That is about the size of the ovary. All right, so here and we have the fallopian tube, and you know that but meantime, what was doing the spermatozoid? The spermatozoid is trying to get here in the uterus, right? So the spermatozoid is coming all the way here. The ovum is here, and the ovum is being released and walking slowly to the fallopian tube, and they are going to be in this moment. But the spermatozoid is coming here. It's like a few of them because mostly are being killed, slaughtered in the pathway. But again, one mistake. Some of them are going to take this way, wrong way. Goodbye. So the other group are going to come here, and they are going to find the ovum. This is the ovum, and this is the spermatozoid are going to be found in the ampulla. This is the most common. This is the most common place. How long is taking? 1 day. From here, the spermatozoids only have 24 hours, and they are going to, well, the ovum too, and they are going to just find each other. Okay. After this, you count in 1, 2, 3, 4, 5, 6, and then 6 days after fertilization or fecundation. Fecundation is the same to say fertilization, and they are going to get into after 6 days into the uterus. All right, so we can talk more about that when we are doing anatomy physiology. All right, so that is what I want to remember. All right, so we finish lecture 11 and lecture 12. If there is some comments or some questions, please, this is the moment to go and do it. We are going to enter in the Bioscience lecture 13 and 14. Okay, I'm going to make the microphone on. So I would like to see somebody have some questions. Just a moment. Alba, you have a question? Everything is okay? Why is not muting? Okay. All right, so I'm going to unmute some, let's see. All right. David, can you hear me?
Yeah, I can hear you.
Any question? Everything is okay?
Everything's good, sir. Everything's good.
All right, great. Okay, so let's start. So there is no raised hand, so let's start with the next chapters are going to be 13 and 14. Okay, so more genetics. All right, so this is something that we already talked. I'm going to just remark about what is important for our next activity. Okay, so you already know about phenotype. We have homozygous, we have heterozygous. Remember, homozygous is we have identical alleles, right? So now makes sense a little bit more, I guess, hopefully. Alleles are the comparison of two locus, right? Two same locus of the, let me see here, let me see, just a moment, please. This pen is not really friendly. Okay. No, I cannot do better. Okay, perfect. Okay, that's it. So we have homozygous and we have heterozygous, right? So homozygous is when they have two alleles that are actually the same, blue eye and blue eye, right? Heterozygous is going to be different. So what is important about, and I'm sure you are actually very aware about this, we are going to do some Punnett square. Punnett square are going to be coming most likely. So please, I want you to be sure that you are practicing your Punnett square. Just a moment, something is wrong. Okay. Okay, so one thing I want to remark here is this. For example, you have a father who has blood serotype O, and the mother is serotype A. Okay, so you need to play with your options, right? If you have somebody who is O, the the, this person, so this person, okay, is OO. It should be OO, right? It cannot be other options. So the parents, this is the parents are going to be, so this is the parents. Let's make it like this, okay? And this is the parent, the father. The father should be giving to this kid O and O, correct? They cannot give it other ones. So that means that when you have O, you have this option on. But when you have A, so let's put it this A. Let's make a girl right now. So this is A. This A could be two options, right? It could be homozygous AA, or it could be heterozygous AO. So this is still having serotype A, and this is going to still having serotype A. So you need to just be careful when I'm not going to ask you. If I'm telling you that this girl is homozygous A, that means that is AA. But if I tell you that is heterozygous A is going to be A and 0. Is that clear? Right? So based on that, you need to create your Punnett square. If I ask you, pay attention in the homozygous and the heterozygous. This is actually very important, okay? All right, so let's, we make a big shortcut there. If you want me to go over again, please let me know. All right, so let's go here. Let's keep going. Okay, we was talking about hemophilia, and we was talking about the color blindness, right? So that is something that we already can pass. Okay, so give me a second. Something is in the machine not doing right. Just a moment. What is this? Oh, okay. All right, so let's talk about the eye. Okay, talking about the eye here. This graphic is going to help us to make the. Remember, I'm doing important remarks. So everything that I remark is actually very important to keep in mind all the time. Okay. All right. So here we have the eye. We have here is the, what is this? This is the sclera, right? That is the sclera. Sclera. So I'm sorry, my marker is not that good. Sclera. This is so the sclera is going to continue anteriorly, and here's something happening. So it's going to continue, and then anterior to the eye, now it's going to be called cornea. But here in this portion is where the transformation is going to happen. So this is going to be white, as everybody knows, but here is going to start become clear, transparent, and that is what we call the cornea. The cornea is epithelial tissue, and the cornea have 5 layers. If you damage more than 2 layers, that is going to be a scar. Can you imagine having a scar in front of your eye? Okay, so be careful on that. So we have the sclera is the outer layer, The middle layer is going to be the choroid membrane. It's red, very suggestive. It's red, okay? And the internal is the retina, internal. So medial is choroid, internal is retina, and the outer is going to be the sclera. I want just you to pay attention to this. The optic nerve is cranial nerve number 2, right? 2 eyes. Cranial nerve number 2. This cranial nerve is going to, if can you see here, it's going to open like a flower, all the way lining the internal, the surface of the eye, and that is called the retina. So you see the retina, all this is the retina. So what is retina actually is the continuation, the projection, the elongation of the optic nerve coming into the eye. Okay, so it's covering the inner cavity of the eye. All right, so that is what we need to say about the three layers. The eye is 3 letters, eye 3 letters, eye 3 layers, eye 3 chambers. So everything is 3. Okay. All right. So the chambers. Chambers, we have to recognize all the structures first. We have the cornea. We have the iris. This space, please use your imagination a little bit. Here we have this space is the pupil, the dark area in the eye. Okay. And this pupil is surrounded by the iris. Posterior to the iris, we have the lens. We have the lens or the crystalline. And behind the lens, we have the vitreous body. The vitreous body or the vitreous humor. So it's called body or humor, doesn't matter. Okay, it's the same. So if you see here the consistency, well, the draw is very good here. It's kind of more viscous, right? More viscous, more like a gel. This is going to help to keep the shape of the eye, number 1. All right, so we have the 3 chambers. The 3 chambers are going to be the anterior chamber. Where is anterior chamber? Here. All this is anterior chamber. All this is anterior chamber. Anterior chamber, anterior chamber, anterior chamber. This anterior chamber is between the cornea and the iris. Simple, right? Then we have the posterior chamber. Posterior chamber. Posterior chamber is this. This is the posterior chamber. Please. This is the posterior chamber. Posterior chamber. That's it. Posterior chamber. And the posterior chamber is in between the iris and the crystalline, right? Or the lens. This is the lens. And this is the posterior chamber. Posterior chamber. Not this. This is the posterior chamber only. Period. Here is the posterior chamber. Behind that means behind the most from the lens, that is going to be the vitreous body or vitreous humor. Okay, so we have 3 chambers: anterior, posterior, and the vitreous body or vitreous humor. So you already know that if you have high production here in the posterior chamber, we produce the aqueous humor. The aqueous humor. Aqueous humor. The aqueous humor is produced by the posterior chamber, and where is going to go this fluid? This fluid going to the anterior chamber. So all the yellow I put here, all the yellow I'm drawing here, that is the flow of the aqueous humor. Aqueous humor. Aqueous humor, they don't go here. They do not go here. They go between the anterior and the posterior. That's it. When you have too much fluid here, we are going to talk about glaucoma, right? When you have too much fluid, you have too much production of this fluid, or too low excretion of this fluid. We are going to see that later, another time. that fluid is going to accumulate, and produce high pressure on the eye. And the vitreous body is pushed against it's going to push the optic nerve. So when they push against the bone, because this is bone, they are going to cut little by little, fiber by fiber, the retinas of connections to the optic nerve. And that is going to then end can lead into blindness. We're going to get into blindness. Okay? All right, so let's keep going. So that is about what I want. Oh, just pay attention. The lacrimal gland, please don't tell me the lacrimal gland is here. Okay, the lacrimal gland is here in the upper outer portion of the eye. Okay. All right, so that is about the eye. All right, talking about the cones. So it's very important about this. So cones. We have the retina. In the retina, we have receptors. What receptors are going to be the cones and are going to be the rods. Okay, cones. I said color, right? Cones, color, cocoa. Rods is going to be for night vision. Okay, so we know that. What I want you to remember is about the retinol. Retinol, retinol Retinol is same to say vitamin A. Vitamin A. Okay? You can call the retinoic acid vitamin A or retinol. Okay? So this is part of the, remember FADEC is the fat soluble vitamins. So that's why you have creams that you we put in our hands, right? Because it's A. A is FADEC, it's fat soluble. So that's why we can put it in the skin. Can you find some cream with vitamin D? Yes. Can you find creams with vitamin E? Yes. Vitamin K, sometimes they are not selling because they need to be under prescription. But do you find creams with vitamin B? Right? You can find some vitamin C once in a while, but not that common. But mostly it's A, D, and E. Okay? All right. So one thing I want to tell you here is that the retinol is useful for the rods. Same vitamin A, retinoic acid are using for the rods. Okay, so what I'm trying to tell you is that if you take vitamin A for your eyes, right, to have better vision, to see better colors, you will never have results because that is not going to affect the cones, only the rods. The rods is for night vision. Okay, period. All right, so that is about the rods. We have the choroid. The choroid, remember please, choroid is the middle layer of the layer of the eye, and that has a lot of vascularization. Please, vascularize. It's a lot of work. This is medical terminology. Vascularize. That is going to have high number of vessels. That is what means vascular, highly vascularized. Lower vascularized means a lot of vessels. Okay. And that is going to give, obviously, can give blood supply to the sclera and to the retina. Okay, perfect. The conjunctiva, remember we was talking about the conjunctiva. This is the eye, this is the iris, whatever is the front of the eye, and this is the eyelid, another eyelid. This is the eyelid, and the conjunctiva is a membrane, right? It's a membrane that's coming here, and it's going to just reflect all over here around the eye. So that membrane is going to be called the conjunctiva. Okay. All right, so here we have the macula. Macula is a small indentation. Okay. Macula, if you see how the lights are coming, the light is coming this way, right? Oh, sorry, this way. So it's going to be close to the, it's not directly to the macula, but it's close to the center of the eye. So this macula is where we have the higher number of what cones and rods. And the macula is like this. And here in the center, we have, all this is the macula, sorry, all this is the macula, but the center is going to be what we call the fovea, is the indentation that you see here. So macula, what is macula? Macula is here, a spot. I'm drawing somewhere here, just a moment, spot. Okay, and let's make a spot here. And when you go and see at the level of the macula, you see the macula like that. But if you see at the level of the macula, you will see like this. This is called the fovea. The innerst layer, the innerst portion of the macula is going to be the fovea. That is, even though here we have macula, high concentrations of cones and rods, the fovea has higher concentrations of cones and rods. So as you go, they go more deeper, the more, so here we have cones and rods, but here is where it's going to be the highest concentration. Even more, in the center of this indentation is where we have the highest concentration of rods. Okay, so we have intrinsic and extrinsic muscles. Intrinsic are going to be the iris, okay? The iris, the pupil, right? The pupil, this is the pupil. The pupil is going to have muscle, and this muscle can be circular too. They are going to contract and relax, and they make the pupil increase in size or decrease in size. So that is called meiosis, when it's very narrow, and mydriasis, when the pupil is very dilated. Ciliary muscles, I just want to show you here, ciliary muscle. So this portion, can you see this? This is the anterior chamber, posterior chamber. Here is the ciliary body. But we was talking ciliary muscles, right? So we are going to see that. Ciliary body. Ciliary body. Ciliary body is the complex, is the building, let's put it this way. All this is a building. Part of the building, they have some elevator, whatever, right? So the part of the building. So we have here the ciliary body is composed by ciliary muscle. This is the ciliary muscle. Can you see these lines? Can you see here? Ciliary muscle. The ciliary gland and the ciliary vessels. Ciliary gland here is what is doing this portion, producing the aqueous humor. The aqueous humor. That is what is doing the ciliary body. All right, so let's finish with the eye, I guess. If there's any other question, just please let me know. Let me see what else we have. Just a moment. All right, so let's go to our next topic. I think it's 14. If there's any question from the previous, very high yield topics here is first of all, what is disease? When you're talking about disease, you're talking about impairment of normal physiological function. Your functions are actually, or the organ are going to be impaired. So we need to be careful on that. Infection disease is the entrance, is like invasion, enemy, growth and multiplication of the microorganisms. All right, so we talk about that. Here we have our friend E. coli that is moving there. This is actually a very good cartoon of the E. coli. So prokaryotes. So prokaryote, who is a prokaryote? Prokaryote is a bacteria, right? Bacteria. And the eukaryote is EU, is our cells. Our cells. So you and I, we have eukaryote cells. The bacteria have prokaryotes. And you remember the prokaryotes do not have nucleus. The eukaryote, we have nucleus with the DNA. So this is DNA of the prokaryote. That is just like in a soup. It's everywhere. Meantime, the eukaryote have an organized manner, the DNA inside the nucleus. So, all right, so let's keep going. Okay. So the classification of the bacteria is, remember, who is more difficult to kill is the anaerobic, right? The aerobic is going to be having It's more easy. So it's going to, just remember, gram positive, sorry. Gram positive, one layer. Besides the plasmatic membrane, right? And the gram negative is double layer. Remember the castle with a double layer, right? Double wall. So anaerobics are going to be more difficult. Just remember, gram positive are going to be, gram positive is going to be what? Where's my, sorry. Okay, I cannot see. Okay. So gram positive is going, and gram negative, right? Gram positive is blue or purple, whatever you want to see, because you know that people have different perception of colors, right? It's slightly different, are not the same, the blue from one to another blue. Anyhow, so gram is going to be red or pink. So you already know the mnemonic here for the gram positive, right? And for the gram negative. Okay. All right, so let's keep going. So we have the shapes are going to be basically a coccus. Coccus, what is going to be useful for you, always remember this, is coccus and bacillus, period. That's all what I want you to remember. That's what is going to just keep it in your mind. Always they're going to help you that. Okay, oh, the flora. Okay, so if you see here, this is the mucus of the intestine. Here we have the flora. These green guys are going to be our friends, bacteria living in the intestine. And what they are doing is just waiting. Waiting for what? For the fiber. Fiber is coming. So you want to eat vegetables, salad. And that is, this fiber is going to be grabbed by hugging by the bacteria. They are going to eat it. They have all the nutrients from the cellulose. The remnants are going to actually be called fiber. Fiber is going to produce the stools, right? It's going to give the shape of the stools. All right. So in addition to that, the bacteria doing that, they are satisfied and they are going to produce some substance that we call the vitamin K and the biotin. Biotin, okay, biotin is the vitamin 7, B7. So all that are, this is vitamin B and this is vitamin K. Okay, so that is our source, important source of vitamin for us. So that is, remember this word, symbiosis, symbiosis, symbiosis. Symbiosis is a mutual cooperation, mutual beneficial relationship. So we help them, they help us. Okay, so that is what we call the normal flora. So please be careful on that. All right, opportunistic is when the immune system is going down, right? So the bacteria is waiting for you in order to, waiting to immune system go down in order to attack. Okay, talking about the ear, we have, well, it's true. We have 3 letters again, 3 letters, and we have the outer, the middle, and the inner ear. So if you have, let's make a picture here, there you are. So let's make a shortcut and a summary of what about ear. All right, so we have, this guy is called the, just a moment. I have problems with the pointer and with the marker, so I don't know how to make it more thicker. Only just this. Okay, so here we have the pinna. Pinna is double N, I guess. Pinna, okay. This is the auricle, pinna or auricle. Okay, we have here, this is the helix. Helix. Here is going to be the antihelix. Here we have, well, it's not seen here, but the tip on the front is going to be tragus. And the small tip here is going to be the antitragus. So that's it. So there's more parts, but it's the only thing we need to remember. So we have the pinna or auricle, and there are going to be the outer ear. This is the outer ear, the outer ear, and the outer ear go from where? From the pinna or auricle to the ear canal up to here, including this. So all this is in the outer ear. This is the outer ear. Okay, outer ear. All this is the outer ear, including that tympanic membrane. So then we have, what else we have here? Nothing else, right? So we have some small glands here that are going to produce some wax, right? The wax that is producing. This is the tympanic membrane that is like a membrane, like it's like a vibrate membrane. So here we have then the middle ear. So if you see here, this is like a sponge. These guys are this is a bone. Is the temporal bone. Is the temporal bone. Temporal bone. This is the temporal bone. All right, so temporal muscle, etc. So anyhow, so here we have the temporal bone, and inside the temporal bone, so can you see it's perforating? Imagine that the temporal bone is all this. So the ear canal is practically perforating the temporal bone, getting drilling inside, and they are going to make a cavity where it's going to be established whom? The middle ear. In the middle ear we have we have mees, remember mees? Mees is the malleus or hammer, the incus or anvil, and the stapes of the stirrup. So this is the malleus, this is the incus, and this is the stapes. All right, so that is this muscle. This is the malleus. All this is the incus, and this small one is the stapes. So what you're doing is amplifying 20 times the sound wave. Sound wave is coming this way, like this. Sound wave, sound wave, sound wave, sound wave. Boom, 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 and they are going to increase the sound waves 20 times. Now, let me go back here. Oh, I cannot. Okay, let me. So now that we have that, we are going to talk about the inner ear. All right, talking about the inner ear, we have three main components, right? Three main components in the middle ear. So what are components are those? Are going to be is going to be here the vestibule. This is the vestibule. So again, so it's inside the temporal bone. We have the vestibule. This is the vestibule. Then we have the semicircular canals, semicircular canals. Can you see semicircular canals? And here we have the cochlea. So we have three structures. All right. So we have, oh, by the way, it's interesting, right? So we have three in the outer ear, the auricle, 1, ear canal 2, and the tympanic membrane 3. We have in the middle ear, we have malleus 1, we have incus 2, and stapes 3. And here in the middle ear, we have the vestibule 1, we have the semicircular canal 2, and the cochlear 3. So we have three of these. Well, we need to add here in the middle ear the eustachian tube. The eustachian tube. I almost forgot about the eustachian tube. The eustachian tube is a communication of the oropharynx, or let's call the pharynx better, pharynx, with the what? With the middle ear. So the bacteria or virus is coming here, climbing, climbing, jumping, 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 and they are going to get into the middle ear. That's what produce otitis media, inflammation of the middle ear. And I was showing you, if you push the tragus and you trigger out a lot of pain, that is a sign of otitis media. All right, so let's keep going. So we have this in the cochlea, we have the Corti organs, who is going to detect the vibration. So all this is fluid. Here inside, this is air, but here in the inner ear, can you see inside is fluid? Inside the cochlea, inside the semicircular canal. And outside there's a space here. That is fluid too. So everything is surrounded by fluid inside and outside. That is the perilymph. But here, the fluid inside is called the endolymph. endolymph. And the outer is going to be called the perilymph. Perilymph. The V didn't mention because I just we want to focus on the endolymph that is inside. So vibrating, they're going to stimulate the corti organs, and that is going to make, remember, the mechanical forces that are going to produce electrical forces. So that is what is happening here with the corti organ. That is the transducer, let's put it away, the corti organ, that is going to allow the mechanical forces of the wave pass into electrical impulses into the vestibular cochlear nerve. See, cranial nerve number 8. Number 8. You write 8, looks like an ear. So it's 8. And what is the name? Vestibular cochlear nerve. That's why it's called vestibular cochlear nerve. Simple to remember. All right, so let's keep going. All right, so we finished, I think, the this part. Let me finish this. We're going to discard. So we have 13 and 14. So let me move about this. So we are going to do 15 and 16. All right, talking about 15 and 16. All right, so just a moment. Okay, so, oh, by the way, we need to talk about the sporulation, by the way, sporulation, okay? So I need to, the sporulation. The sporulation, so let me see here. Okay, sporulation. We have sporulation, okay? Sporulation are those bacteria who are going, this is the bacteria, okay? And the bacteria then is going to, when they found some bad environment, they are going to cover, in addition to some capsule. They are going to surround it like this. So they are going to be isolated. They are not going to react to anybody until they are going to see that the outer environment is going to be more benign for them. So once that happens, this is going to rupture and the bacteria is coming out again and start to reproduce. This capsule is called the endospore. The endospore. And the sporulation process is involving the production of the endospore, then the multiplication. So sporulation means the bacteria produce endospore and then multiply. That is sporulation. Okay, and endospore is part of that sporulation. Endospore is the cover of the bacteria to protect himself. Okay, so we divide that. We was talking about toxins and the toxins. Remember the toxins. Toxins are going to be, for example, the toxins of we have toxins that produce damage of tissue damage, right? Tissue damage. So we explain a little bit more in class about this. So if there's any question, we can go ahead another moment. All right. So in order to prevent these toxins or proliferation of bacteria, remember one bacteria reproduce every 20 minutes. If you have 1 million bacteria, in 20 minutes you have 2 million bacteria in the 2 million bacteria in 20 minutes. So they're going to multiply. So here what we need to remember is cooking kill bacteria. Frozen slow down bacteria. They are not going to kill them. They are going to be frozen. They are going to decrease the speed of multiplication. Then we have here we have the the temperature that we need to keep the food less than less than 40 Fahrenheit. That is, or more than 140. So it's going to be 40 to 140, right? Should be more than 140 or less than 140. This between 40 and 140 is the dangerous zone. Dangerous zone, and we don't want that. Okay, you can reheat the food one time. Okay, so I know that we do different stuff, but this is what is recommended, and that's what we are going to ask. Okay. All right, so the range is 40 to 140 Fahrenheit, and in between, so that is the danger zone. So 41, 42, 100, 139, those are danger zone, but danger zone. But if there's more than 140, you're killing bacteria. If you are having less than 40, you are making the bacteria diminish in his speed of reproduction. So when you reheat the food, you reheat only one time, and when the food is out of the when the food is out of the when you're not eating, the food can be out 3 hours total outside. Okay. For example, you take it out half an hour. Right now, 2 hours later. So actually, that is the top. So 3 hours total. Okay. Okay, so let's keep going. Oh, this, please pay attention to this. This is very high yield for our examination. Okay, E. coli. E. coli cause severe diarrheas. That is, this is actually our friend E. coli here. Well, it's not. It's good friends sometimes, but it's not all the time. E. coli is going to give a lot of UTIs. It's very common to produce UTI. Why? Because these guys mostly are in the stools. Stools. Okay, so diarrhea. So then we have Staphylococcus aureus. Staphylococcus aureus, they can produce, just remember the potato salad, very common potato salad. The potato salad, we have this, you leave it on the outside for more than 2 hours, the salad start to heat it up, right? Warm the salad, and that is the area where the bacteria start to proliferate, and that is going to produce an enterotoxin, and this enterotoxin produce inflammation of the intestine, is going to lead into nauseous, vomiting, and diarrhea, especially diarrhea. So nauseous, vomiting, diarrhea, potato salad. So, well, not all potato salads. Of course, you need to keep the temperature and no more than 3 hours outside, right? Okay. So we have the MRSA. MRSA mostly is common in the bed sores. Treponema pallidum, syphilis. Syphilis. Can you see these gummas? Those are lesions. If you touch, it's like a very tender, very tender. There is no sensitivity, there is no painful, but it's tender. And here we have these lesions. This is all over the body, all over the body. This too is all over the skin. And that is going to, if you push here, they're going to be like pustules with crust that are not painful. Okay, that is syphilis. So we have the gonorrhea. Gonorrhea is a cause of excessive secretions. So this is a lot of pus. What is pus, by the way? Pus is going to be the is water plus dead red blood cells plus dead white cells. Basically, is that plus dead bacteria plus dead, we have especially the macrophages. So pus in general is a cocktail of all these guys. Okay, water, red blood cells, white cells, bacteria, macrophage. All right, so let's keep going with the Haemophilus influenza. And for Haemophilus influenza is, again, please don't be confused with the flu. Flu is different. This is flu virus, influenza flu virus is the virus, but this is totally different. This is Haemophilus influenza bacteria. So it's bacteria. This bacteria, mostly what is given is respiratory infections. They can lead into Meningitis. What is meninges? Meninges, this is the brain. Let's make the brain like this. This is the brain. There is a cover around the brain. One cover, another cover, and another cover. So 3 layers of covers. So that is called the meninges. The meninges. And this guy can lead into meningitis, inflammation of the meninges. So very, very painful. It's going to irritate the, it's what we call encephalitis. 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 Okay, and meningitis. Okay, so we have salmonella, we have diarrhea, fever. So this is what you find in the chicken, and that is, well, not all chicken, of course, but you need to be careful with that. And the salmonella, they like to live in this, coming from the farms of the chicken is being accumulated in stools all over and that stools are the salmonella that can be transported on the meat or over the eggs. So the eggs have a process that they are going to clean it from outside before they sell it to the store. So so be careful just to clean very well your products. Symptoms: diarrhea, fever, salmonella. So with the fever, we have breakouts of salmonella every certain time. The last time was about 20 years, but it's going to appear. Nobody knows when it could be the next one. All right, so let's go to Bacillus cereus. Usually they're going to do the nausea and vomiting, but no diarrhea. This is mostly located in the soil, and they go especially in the next day rice. So if you leave the rice out 1, 2, 3 days, these guys they love our rice too. So what they're doing is to proliferate, and when you eat it, you start to have actually nausea and vomiting. There is no diarrhea involved here. Okay, coming from the soil. All right, so Helicobacter pylori, you see here the helicopter here, just to remember, is going to produce peptic ulcers. Peptic ulcers. Mycobacterium tuberculosis produce the tuberculosis. Okay, so Clostridium difficile. The Clostridium difficile is one of the bacteria who sporulate, correct? Okay, so one of the bacteria that are going to sporulate, and they are going to be most common cause of nosocomial infections. Common causes of nosocomial. Nosocomial means hospital, right? So remember, please, I want you always remember that these guys, when you have this, you will never forget, I'm sure, by the smell and by the color and by all that. And the precautions, safety precautions, you need to always wash your hand with soap. Soap. No, alcohol is not enough in this case. Okay? Soap. All right, so Clostridium prefringens, very important, please, open eyes, open ears. They are going to sporulate, by the way, these Clostridium prefringens. Mostly are being located in diabetic patients, okay? It can be caused by, it could cause gas gangrene. So that necrosis, when you see dead tissue are going to be having a black color. Okay, it's going to be dark. Clostridium prefringens. Then let's see another Clostridium, and we have the Clostridium tetani, or Clostridium tetani is going to produce spasms of the whole body, and these spasms including the diaphragm, so you cannot actually breathe. So spastic paralysis. Spastic means contraction, spastic contraction. Then we have the Clostridium botulinum. Botulinum is the opposite, flaccid paralysis. Flaccid paralysis. Flaccid paralysis. That is mostly related when you have some canned food, some can that is being damaged. Okay, so that is about that. Just remember about the viruses. The viruses, we have two types of viruses. We have virus DNA virus, and we have the RNA virus. Examples of DNA virus, we have hepatitis and what herpes, right? And example of hepatitis RN virus are going to be the HIV virus. Okay, prion is a protein mostly located, for example, in the mad cow disease. It's a protein that cannot be denaturized, so that means destroyed by heat. It's not going to be able to destroy by heat. So people need to eat it, uh, these organs, in order to have that transmit the disease. All right, so prion. So prion is not a living organism, right? It's a protein. Okay, virus is not a virus organism. It's not a living organism, the virus. Okay, so let's keep going. Talking about the bones. The bones, please, I want you to review this chart 2-3 times, and that is actually review about what I want you to see about bones. All right, so how many vertebrae we have? 33 vertebrae, 7 cervical, 12 thoracic, 5 lumbar, 5 sacral that are fused, and the coccygeal are going to be 4 vertebrae that are fused. We was talking about the ribs. The ribs, remember the ribs, we have, the ribs are going to be true, false, and floating ribs. The floating ribs are per 11 and 12. 1 to 7 pairs are going to be the true ribs, and the false ribs will be 8, 9, and 10. Okay, so those are the false ribs, and the floating ribs 11 and 12, the last two. Okay, so the difference between them is the floating ribs anteriorly are not attached to anything, the floating ribs. Then we have the false ribs attached to the sternum through a cartilage, and then we have the true ribs attached directly to the sternum. All of them correspond, all the ribs are going to correspond to one thoracic vertebra. All right, so let's go to the blood now. Okay, so in blood, so I'm going to make a summary about the blood. There is a lot, as you can understand, about what is blood. So we are going to make just a summary. So number one, what you need to remember is this. First, blood is going to be composed by plasma and form elements, period. Form elements will be red blood cells, white cells, and never forget this, please. Thrombocytes is the same to say platelets. Please, platelets is the same to say thrombocytes. Okay, thrombocytes, platelets. So those are the 3 form elements, 1, 2, 3. And these form elements are coming from the what? From the red bone marrow, right? Red bone marrow. And these all, when they produce all these form elements, is called the hematopoiesis. This formation of red blood cells alone is going to be called the erythropoiesis. Erythropoiesis, red blood cells, plus white cells, plus thrombocytes. So the formation of thrombocytes or platelets plus white cells plus the erythropoiesis, means the red blood cells, all together is called the hematopoiesis. All right, so I think it's clear on that. All right, so then we are going to see that we are going to talk about the hematocrit. Hematocrit, very simple. We have a tube, remember. We have the blood, and the blood is going to, this is 50%, this is about 45%. These are red blood cells, and this is plasma. Plasma is 55 of the volume of the blood, and RBC is about 45% of the volume of the blood. So what here in between we have the white cells and platelets, and here in the plasma what we have 55% of all, we have water, we have proteins, important to know, the albumin, the clot factors, we have the clot factors including the fibrin, right? Including the fibrin. So we have very important proteins, carbohydrates, lipids, vitamins, minerals, hormones, enzymes, etc. Right? That is the plasma. All right. So let's keep going. All right. So the pH, you already know, function of the blood, transport substance, immune system, stabilize blood pH. The normal value of pH blood is 735 to 745. We have, they are going to stabilize body temperature. We have a vasodilation and vasoconstriction. Okay, so a class of hemorrhage type 1, type 2, type 3, and type 4, right? So you just need to remember that we have less than 1 liter, less than 1, less than 2 liters, less than 3 liters, okay? And this is more than 3 liters. So yes, if you want to remember that way, it's going to be. So 10 cc or more or less is actually not big difference. But definitely, yes, you need to remember that we have 4 classes of hemorrhage, okay? All right. Okay, albumin, the value of albumin, please, 3.5 to 5. What is hypoalbuminemia? Low amount of albumin in blood. So what means if you have low amount of blood of albumin in blood, there is nobody who retain the water, and the water starts to escape, and you start to have edemas. Okay, hyperalbuminemia, the opposite. If you have, for example, high levels of albumin, that is giving when you have a patient who is dehydrated, they're giving this albumin. But if you give too much albumin, too much albumin is going to retain too much water in the vessels. That is going to be the opposite of dehydration. They can they can lead into flooding of the lungs, for example, pulmonary edema, cerebral edema. Phobia and the edema, we have 2 types of edema, right? The pitting and the not pitting. Okay, the pitting edema involves veins, especially veins, okay, capillaries, but the not pitting edema involves what? The lymphatic vessels. So that is the big thing that you need to remember, lymphatic vessels. Not pitting, lymphatic, pitting is the veins. So escaping of fluid out of the veins. And the lymphatic do not keep the function, is an obstruction of the lymphatic. Okay. Sodium, please must remember this. Sodium 135 to 145. All right, so we already talked about the platelets. We have the coagulation. Remember, clot. What is a clot? Here's a clot. Clot is a fibrin. What is a fibrin? Secondary coagulation. What is platelets? Primary coagulation. We talk about that. Secondary coagulation. So we have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 12, 13 proteins that are going to react one after another. And what is the product? At the end is going to produce fibrin. So hemophilia, we don't have one of these proteins. Hemophilia, we don't have one of these proteins. So they are not going to produce fibrin. So that's why the patient is bleeding easily in hemophilia. So we have certain proteins that react one after another. They are going to, as a final product of the reaction of these 13, one after another, is the fibrin. So that is a clot. Okay, clot is the same to say thrombus. And it's very suggestive, the name, because it's coming from thrombocytes, right? Thrombocytes are platelets. Okay, so we have that. All right, so talking about the arteries and veins, so we need to remember very important few things here. We have artery, we have vein, we have capillary, and we have the lymphatic. Let's add the lymphatic here. Very simple. Artery, we have 3 layers. 3. Vein, 3 layers. Lymphatic, 3 layers. Everything is 3. Everything is 3. Then we have the capillaries. Well, not everything. Capillaries is 1. Okay. 3 layers of the artery. We have the tunica intima, tunica media, and the tunica adventitia. Okay. Intima, media, and adventitia. And the vein, we have the tunica intima. See? Tunica intima. The tunica media is very thin. That is the difference between vein and arteries. And the capillary, they have only just the tunica intima. The tunica intima. The lymphatics are going to be similar as the vein. They're going to have tunica intima, tunica media, and the what? And the adventitia. So here we have bulbs in the vein. We have bulbs in the vein. We have bulbs in the vein. Bulbs in the vein. In the arteries, do not. In the capillaries, neither. So these are similar to each other. Okay. So that is about. So you compress. If you compress the artery, they are going to be same shape. But if you compress the vein, that is going to collapse. So that is important for the circuit, so the venous to the for the venous return of the blood to the heart. Okay, so let's finish this. Sorry, it's gone. So we finished 15 and 16, I guess, right? Yeah. Let's go to the 17, 18, 19, and we finish with a review. Yeah, I know that some of you are questioning what about the 20 questions, right? So we can do, I will do it at the end. All right, so let's talk about the red blood cells, hemoglobin. You know, hemoglobin, where is the hemoglobin? How? Okay, just a moment. Okay. So this is the hematocrit. So I need to get more familiar with these pointers and all that. Just a moment, please. Give me a second, please. I don't know what. Okay, so I cannot make it thicker. I don't know how to. Okay, perfect. So we have red blood cells. We have the other name is erythrocytes. 120 days. They're going to die in the spleen, right? In the spleen. Remember the red blood cells. They release the hemoglobin. The hemoglobin is going to release the bilirubin. Okay, and the bilirubin go to the bile. Okay, so the red bone marrow produce 3 lines. The red bone marrow produce white cells, produce red blood cells, and platelets. All right, so hemoglobin plus carbon dioxide is the carbamino hemoglobin, and hemoglobin with carbon monoxide is carboxyhemoglobin. That is when you have toxic levels. Then you have here in the normal hemoglobin, when they transport oxygen, it's the oxyhemoglobin, that is normal. Deoxyhemoglobin, that is normal. But when you use abnormal, you have high levels of carbon dioxide, is toxic carbaminohemoglobin. Hemoglobin with carbon monoxide, that is actually, listen, is considered permanently attached to the hemoglobin. Okay? This is kind of name, but the different is attached 4 hours. So if, that means it's forever, because after 4 hours, if all the blood is being saturated with carbon monoxide, there is no way. So the person will die. All right, so let's go to anemia. Anemia, female 12 to 14, male 14 to 16. Signs of anemia. So please, fatigue, obviously, please, just to think about it. If you have anemia, you have less oxygen that is carried by the blood. So you need more oxygen. So when you are tired or exhausted, you need to oxygenate more your lungs, and you cannot because you have low number of red blood cells. So that is going to produce fatigue. Iron deficiency, we already, oh, okay. Microcytic hypochromic, so please 5 times this, okay, write down this 5 times. Microcytic hypochromic. The best absorption is going to be meat for iron. Meat, okay, meat. We have vitamin B12. Vitamin B12 is, they need the intrinsic factor. Remember the intrinsic, oh, by the way, before that, iron needs acid. Acid. So keywords for iron. Acid, best source, meat, microscopically is going to be microcytic hypochromic, period. Vitamin B12, we have intrinsic factor that is what? Is a protein produced by the stomach, and the intrinsic factor, they need to be attached to the vitamin B12 in order to be absorbed. Otherwise, they are not going to be absorbed by the intestine. Okay? Vitamin B12. Mostly, what it's going to do is that megaloblasts or megaloblastic anemia. Megaloblastic anemia. Megaloblastic anemia. The other one who produce megaloblastic anemia is vitamin B12 and the folic acid. The folic acid are going to produce megaloblastic anemia too. So that is folic acid is the vitamin B9. Okay, vitamin E, important please. That is going to sweep out what? The free radicals. So if the red blood cell is here, the free radicals are trying to attack the red blood cell. So super vitamin E is coming and is going to take it out, all of them. And the red blood cell is going to last longer because it's not going to be destroyed by the free radicals. So if you have low levels of vitamin E that is not that common, they are going to produce hemolysis. Okay, hemolysis is break out of the red blood cell. We have another highly topic, please pay attention to this. That is going to be the hemolytic anemia. We have, for example, what we have, the sickle cell, right? Sickle cell disease that is going to produce hemolytic anemia. Another one is malaria, hemolytic anemia. Okay, here we have malaria, sickle cell. Okay, deficiency of vitamin E. 1, 2, 3 potential things. Okay, malaria, sickle cell anemia, and vitamin E. All right, so we was talking about malaria already. Remember, malaria is transmitted. The disease is caused by Plasmodium vivax. I'm going to go in malaria. What is malaria? Cause is Plasmodium vivax. Oh, here you are, Plasmodium vivax. And the transmission is by the mosquito. So be careful with those tricky things, semantically talking. So that is the transmission versus the cause, okay? All right, so let's keep going. We have the, let me see. All right, so sickle cell anemia, any type of hemolysis, what means that there's a lot of red blood cells are going to die, are going down. And the spleen at the beginning is this size, is the cemetery, and the cemetery is having a lot of work, so they are going to start increasing the function, and the spleen is going to enlarge, and that's called the splenomegaly. So in malaria is going to happen, sickle cell anemia is going to happen, in any type of hemolysis is going to happen. We have jaundice. Why is coming the jaundice from? The jaundice is coming, red blood cells die, they go to the spleen. The spleen is, in the spleen, the red blood cells are going to release hemoglobin. The hemoglobin transforms into bilirubin, and the bilirubin in excess, they go to the bile, but not every can go to the bile. They start to go to the circulatory system. They go to the skin and produce jaundice, excess of bilirubin. When do we have excess of bilirubin? When you have excess of hemoglobin. When you have excess hemoglobin, when you have excess destruction of red blood cells. Sickle cell anemia, you have, for example, you have liver hepatitis because the bilirubin is going to be processed by the liver, and the liver is being bombed with a lot of bilirubin, cannot handle it. So the rest of bilirubin is going to go to the skin, to the other organs of the, not only inside, not only the skin is yellow, everything is yellow inside. If you, if we open somebody who has bilirubin, you will see that everything is yellow inside. Okay, so we have another thing in the sickle cell anemia. This is the red blood cell, and the sickle cell anemia is like this. It's very brittle, and this going to break down very easy, releasing the 24,000 hemoglobin proteins, but this is multiplied by millions and millions, and the vessels are very tiny. Some vessels, these proteins are going to be blocked, and which vessel is most likely is going to be affected are the vessels who give blood supply to the retina on the cortical membrane from the eye. So the retina is going to start to degenerate, and that can lead into blindness. That is a complication of hemolysis. We talk about malaria. We talk about the sickle cell. All right, so actually those most likely can happen. Hemorrhagic anemia is just hemorrhagic, is just a lot of blood lost. It can be inside or outside. We already talked about that. Internal hemorrhage is going to be what we call in the abdomen cavity. External hemorrhage is going to be outside. Okay, all right. So we talk about the type of serotypes. We are talking about the muscular system. Please remember about the fiber. The fiber is a cell muscle. We have the perimysium. The inner is the endomysium. This is the endomysium. We have the perimysium surrounding the fascicles here. And then we have the epimysium that is surrounding the muscle. So that is about that. Remember, we have two main proteins, the myosin and the actin. Myosin and actin, they are going to interact each other in order to produce a contraction. So not always are in contact. They are in contact only when they are contracting. So this is actin, this is myosin, and this is relaxation. Here we have myosin and actin are going to relate each other. See, they're overlapping now. That is a contraction. And they are going away when they are going to relax. Okay? All right, so that is some main muscles that you can review on that. Here we have the immune system. I think the immune system, what I want just to do here is some definitions. Number one, I'm going to make a list of what I want you to please remember for what is coming. Lymphoid organs: spleen, appendix, thymus, gland, and tonsils. We have that the left lymphatic system is going to drain into the thoracic duct. So the left portion of the lymphatic system is going to drain in the thoracic duct into the subclavian vein, left subclavian vein. And the right lymphatic duct collect 25% of the lymph are going to drain into the right subclavian vein. Okay. So we have antigen, antibody. Antigen is any stranger, any bad guy, bacteria, virus, whatever. And we have antibodies that are the markers. The antibodies, we have the phagocytes that are the macrophages, are the monocytes, and the neutrophils. Those are the antibodies, the phagocytes, sorry. All right, so we're talking about that. Security guard, the basophils who produce histamine, right? Histamine. Monocytes, MMM, are going to be the macrophage, okay? Okay, so let's keep going with this. Active immunity, they are going to give you a portion of the germ that is dead. And passive immunity, they are going to give you antibodies. We have the T cells organization, the T helpers, that where is that? the T helpers, the T, oh, it's not the list here, okay, the T helpers, the T suppressors, the T cytotoxics, and the T memory cells, right? So those are, and what is the brain of everything is going to be the T helpers, right? T helpers, we did that in class, so I hope that is very important. Fever and inflammation, please. Fever is important, yes. Inflammation, 5 cardinal signs. Center of fever, control is the hypothalamus, hypothalamus, hypothalamus. So some definitions coming for the, well, in the future it's coming for your next courses. Fever definition is 97 up to 100, that is the normal value. Bacteremia, presence of bacteria in blood. Sepsis means infection. Systemic sepsis, infection of the systemic circulation. Septicemia, infection of the blood. Be careful because sepsis, septicemia, and systemic sepsis have similar definitions. Sepsis is just a generalized infection. Systemic sepsis is the infection in many systems of the body. And septicemia is when the blood is going, the infection is in the blood. Bacteremia means the presence of bacteria. So you have bacteria, and that is something that we need to live with. Bacteria is already an infection, bacteremia, but bacteremia means an infection that is not that severe. But when you have septicemia, you are talking about a huge amount of bacteria in your blood, and that is life-threatening. Already. Well, any infection is life-threatening, but let's put it, sepsis is more severe than bacteria. All right, so remember the prostaglandins. I love prostaglandins, please. And we need to remember that for this exam and for what is coming later. So where are the prostaglandins? In the cell membrane? Arachidonic acid. The arachidonic acid produces prostaglandins and leukotrienes. So everything is for benefit of the body, right? So you want leukotrienes, why? Because they are going to attract more what? More white cells. Thromboxans is because the tissues are already being repaired and cells are, vessels are damaged. So you have thromboxans who are going to attract who? Thrombocytes, who are the platelets. Prostacyclin is a mediator for inflammation. You want inflammation, right? You want inflammation if you're being attacked by a bacteria or from germ. So you need the inflammation. And prostaglandins are going to be another PGE and PGEF are going to be for pain. Pain that is, to have pain is good, right? Why? Because we can detect where something is wrong is happening. So pain is good. All right, so what? Too much pain, we can control the pain in that case, and that we are going to use NSAIDs. NSAIDs is the non-steroidal anti-inflammatory drugs, as you can see here. Okay, all right, so let's keep going. All right, so autoimmune diseases. Number one we need to remember is, wait just a moment. Okay, this. Arthritis, autoimmune disease, multiple sclerosis, MM. What is damage? The myelin. MM. Please remember that, myelin. Then we have the lupus, that is, they're going to affect skin, joints, kidneys especially. These people die from kidney failure, especially in women after 50s are very common. Myasthenia gravis, another big sin here, please, myasthenia gravis. Because that involves that you already have a concept of what is the acetylcholine and what are the receptors for acetylcholine. And the acetylcholine receptors are called the cholinergic receptors. So that is what this autoimmune disease, myasthenia gravis, are going to affect. The cholinergic receptors. What are cholinergic receptors? Okay, okay, let's put it this way. A receptor is your glass, and the substance is the wine. Okay, so if you have the wine is going to be the acetylcholine, and the glass is going to be the receptor, is the cholinergic receptor. Okay, All right, so lymphatics, we already talked about that. We are going to talk about endocrine. I don't know if you raise your hand, we can do endocrine for this time, so you can review later. All right, so I will do it. So we have, if there is any comment, so please let me know. Endocrine is our last lecture, right? Endocrine system is going to divide in exocrine and endocrine glands. So exocrine, you have a duct. You have a duct. Exocrine, sweat glands, for example, sweat glands. We have endocrine, we have no ducts. For example, the thyroid gland, okay? Hormones, we have two types of hormones, the fat soluble and the, what is an hormone? Hormone is a substance who has a target organ, okay? We have, at this moment, what I want you to remember is the fat soluble and the water soluble. Fat soluble are actually those who are being formed by cholesterol. Oh my God, this is so important, cholesterol. Fat soluble. Fat soluble are going to be hormones that are, they need cholesterol to be produced as an hormone. And those are going to be the estrogen, cortisol, the stress hormone. We have the progesterone. We have the testosterone. We have the aldosterone. These guys are so important in our system. That's why we need cholesterol. And water soluble are going to be all the hormones that are going to be in the pituitary gland. So we are going to see that in a few moments. All right. All right, so here I want just make you remember how we can memorize the I'm going to make a page, so I'm going to make a shortcut for this. Just a moment, please. We are going to finish in a few minutes. Okay, so here we have the hypothalamus. Oh, why? Oh, of course. Okay, just a moment, please. Okay. Okay, so here we have, what's happening? Okay, here we have the hypothalamus. I'm going to be, please follow me, hypothalamus. The hypothalamus is the big gossiper, right? Then this guy is going to connect with the pituitary gland. Pituitary gland. Okay, to make a summary of the endocrine, and we are going to finish with this, the pituitary gland is going to have the anterior pituitary gland, and the posterior pituitary gland. The anterior pituitary gland, we are going to have another name that is the adenohypophysis, but at this moment I want to make a summary. So we have, listen to this, we have G first. It's going to be the growth hormone. We have P, the prolactin. We have T, the thyroid stimulant hormone, that is the TSH. We have A, C, T, H, that is the adrenal gland, corticotropin hormone. And we have L is the LH, is the lutein hormone. And we have at the end F, the FSH, the follicular stimulant hormone. Okay, so please, the anterior pituitary gland, you can remember with this word, GPTAL. We are going to use that in maternity, so it's kind of a coincidence, but definitely it's good. GPTAL, growth, prolactin, thyroid, TSH, ACTH, LH. Okay, and that is, and for our happiness, posterior is going to give only two hormones. It's going to be the oxytocin and the ADH. ADH is the antidiuretic hormone. Antidiuretic hormone is going to prevent to pee, so you don't pee. So you retain water, in other words. And oxytocin, the function is to the contraction of the uterus, uterine contraction, and the milk ejection. Milk ejection, I said ejection. In the other side, we have the prolactin. Prolactin is the milk production. Don't forget that, okay? This is very important to remember. Okay, so that is about the, I think we are going to see that in the next class. We are going to just talk more detail about the endocrine system. So if there is any question, please, I'm going to open microphones. Okay, just a moment. Okay, any question, please? San Juana, do you hear me?
I can hear you, yes.
Okay, any question? All right.
I don't have any questions right now.
Okay. Okay, let me see. Beth, any question? Okay, so raise your hand if you have any question, please. I will going to post the video on in the YouTube. So just wait for me in the next hour. Going to appear is going to appear in my channel, that channel. All right, thank you so much. So any question, just let me know. Raise your hand, so please.
My hand raised.
Oh, hand raised. Where is that? Where is that hand?
Me.
Oh, okay. Yes, I can hear you. Tell me.
On my test, I got, when both of the co-dominants, I got it wrong. It says I checked off when both alleles are equally dominant. What it says.
Yeah.
That one I got wrong. So,
yeah. So this webinar, we are going to close the webinar, and we are going to just by email. I'm going to answer all your questions because
okay.
I'm going to record it, and it's going to appear in the system. So, but I'm really going to be very happy to hear to resolve the problem or to clarify the question. Okay, Mariam.
Okay. Yeah, on note that I just wanted to ask if you have any idea where, this is a weird question, but if you have any idea where particularly we have to study from the first part of the class.
Yeah.
From the
so I will do a review. I will do a review also, first of all, okay, so what we need to know everything about the course, right? Everything.
Yes.
We are going to focus on, I'm going to give you some hints right now, some high topics. What is you consider important for us? What you consider important for nursing is going to be mostly coming. For example, I would like to talk about the metabolism of glucose to understand what are the requirements of the needs of the patient. But just to tell you more clear about that, it's already 1 o'clock, but I know that everybody has to, of you probably going to leave. I have less and less students. So I will go over review on Monday.
Okay.
Is that better?
All right.
Okay.
Thank you.
Arlene. Arlene?
Yes.
Do you have a question?
No, I'm good. Thank you.
Okay. Charlene, you have a question.
I don't have any question, Dr. G.
You're okay?
Yes, Auntie.
Okay.
Thank
Cinderly.
you.
All right. Cinderly, you have a question?
Hi. Yes, I do. Regarding the medical terminology, is it all multiple choice, or are there going to be some write-ins for that part of the test?
Multiple choice is what is the
Okay.
we must use.
Okay, thank you.
Thank you. Diana? Diana, you having a question? Okay. Let's see. Alba, do you have a question?
It's bacteria and bacteria.
Alba, are you there?
Doesn't make sense.
Okay. And let's see. I have Diana. Diana, you have a question, Diana?
Hi, Dr. G.
Tell me.
I was just wondering about the same thing about the first part of the course, if you're
Mhm.
going to do a review
Yes,
about.
we are going to do review on that. I would like to have some time. Well, we are going to start at 10. I'm going to make an announcement. No, it's better no. It's better to start at 10, and we will have time to review, by sure. So after that, we have another review from 2 to 4 p.m. I think everybody's going to be in good shape. Just study as much as you can for this week, okay?
So from 2 to 4, you're also going to review about the first part of the course?
About the whole, no, I'm going to make a general review from 2 to 4.
Okay.
Okay. All right. Thank you so much. So I will close the. There's no more questions. Thank you and for attending to this webinar, and we'll see you Monday. Gracias.
Are you going to be posting it on module?
Yes, it's going to be post in the YouTube.
You're going to send us a link.
Yeah, I will send an email to everybody.
Okay.
Yeah, so for this, that's why if you subscribe, you don't need to do that. You don't need to wait. As soon as I put the email, as soon as I put the video, it's going to give you a notification. But in that, you need to wait until I send you an email. So, but you can subscribe and you can receive the notification right away. All right. Thank you, everybody. I will see you next Monday. Gracias.
Thank you.
Thank you.
Thank you. Bye-bye.
Bye.
Bye.